I want to make sure that today's focus is primarily on phospholipids and the phospholipid bilayer and its role as a selectively permeable barrier. So we'll spend a little bit of time talking about the different types of transport. We'll talk about the importance of this selectively permeable bilayer. And then to finish up, I want to make sure today that I have 5 to 10 minutes to comment on the feedback that you provided over the weekend. Okay, so if I get going, somebody stop me at not 7:40. That already passed. 8:40. Okay. Let's start with the question that I gave you for homework, which was you have a cell, a red blood cell in a solution that has 0.1% NaCl. And inside the cell, typically the concentration is around 0.9%. And the question was, what will happen to the cell, and is the solution hypotonic, hypertonic, or isotonic relative to the cell? So, what I want to start out with is turn to somebody next to you, give them your answer, and then if they disagree, try to figure out what might be wrong with one of those answers. Or are they both correct, but they are better together than they are individually? All right? I'll give you 2 minutes. Go ahead.
I'm pretty sure I said, I don't remember. I said,
Aquaporin here is, let's see if I can draw this real quick. So we'll just kind of draw it like a channel here. And aquaporin is small. And what I mean by that is it only allows molecules through that are about the size of water. At some low rate, it allows alcohols through, ethanol. At some low rate, it allows hydrogen peroxide through as well, H2O2, because there's a similar structure of those molecules to water. And the way that aquaporin works is it's highly charged, or it's highly polar on the inside of that channel. And so as water is moving in, it actually kind of ping-pongs its way back and forth to move into the cell. This porin is specific enough that it doesn't allow sodium and chloride through. Okay. Real quick side note, this actually goes back to some properties that we've talked about. This is not a great image because this aquaporin, as the name would suggest, is a pore. It's a cylinder. So quick side note, hearkening back to things that we've talked about in the past. But this face right here that's interacting with the phospholipid bilayer, is that part of aquaporin polar or nonpolar? Nonpolar? Why? Devin?
Because the H2O is bouncing off.
So we've got this in here. This will be polar because that helps to kind of pull the water through. But on the outside, hydrophobic or nonpolar, because in order for this protein to maintain its stability in the cell membrane, it has to have some reinforcing interactions. And in the case of this one, these interactions are going to be hydrophobic interactions between the phospholipid bilayer and the protein itself. Okay? That's a concept that we talked about in the first couple of class periods, the properties of polarity and hydrophilic and hydrophobic substances. Something we'll come back to. Okay, so we've established water here moves through. It does not require energy. Therefore, what type of transport is this? Passive, facilitated, and those terms oftentimes get used interchangeably. there is a very small nuanced difference. One that is not big enough for us to really worry about too much. But passive means that the actual transport protein does not change shape as the molecule is moving through. Facilitated, on the other hand, that protein actually does, or that channel protein actually does undergo some sort of conformational change as that molecule is moving through. The glucose transporter is a good example of that, where it captures glucose on one end and then changes shape and releases it inside of the cell. Very small difference, but the key point is neither of them require any sort of energy to function. Okay, so going back to the original question, why does water move here but sodium and chloride do not move? Abby.
They would be moving against their concentration gradient.
Okay, so if they were moving, these ones would be moving against their concentration gradient here. But why don't the ones on the inside move outward?
Aren't they like within hydrogen bonds with the water?
That's part of it. Yeah, so the hydrogen bonding would help to capture them. The other thing comes down to how are they transported across? There are sodium channels. There are chloride channels, and usually we think of those as being unidirectional in some way. And not only are they unidirectional, but they require energy to function as well. And so in the case of sodium and chloride, we can get some movement, but it requires energy, and the process is inherently slower than it is for water to move passively into the cell. Okay. When we talk about energy being required, what form are we talking about? Typically, we're talking about ATP, right? Typically, we're talking about ATP. The solution outside of the cell, hypotonic, hypertonic, or isotonic relative to what's inside of the cell? We'd say that it's hypo. So, and this was one area where there was a bit of confusion with the terminology, so I want to make sure that we just go through this real quick. When we say that a solution is hypotonic, what do we mean? Higher or lower concentrations of solute? Austin? Lower concentrations of solute, more free water, as some folks would say. Hypertonic means higher concentrations of solute. Isotonic means same, equal on both sides. The other thing I want to point out here is those terms are inherently comparative terms. So when you're saying that a solution is hypertonic, you need to say compared to what or relative to what. So one of the things that I tried to point out on the answers in the journal entries were if you said the cytoplasm is hypertonic, for example, compared to what? Compared to another cytoplasm? Compared to the solution outside? And yes, I know what you're saying, but don't leave it to the reader to guess. Okay? So this is where some people have been asking for more, like in the comments, some folks said, I want more specific feedback. This is some of that specific feedback. Make sure that you are clearly stating what you're intending to state. So with these hypotonic, isotonic, you need to have the comparison in there. Okay. Any questions on this principle? What about, I know we talked a little bit about this already, but why doesn't Streptococcus mutans or Staphylococcus aureus, 2 gram positives, why don't they burst when we stick them in water? Derek?
The peptidoglycan, the thicker there for gram positive, the plasma membrane from lysine
Good. Good. So that peptidoglycan is so thick, and I'll kind of erase all this real quick, but that peptidoglycan on these cells is thick enough and is reinforced by those tetrapeptides. I'm just going to kind of draw them in here like this to provide additional strength. And that serves kind of like a cage in some ways. That membrane can expand and can push against the wall, but that wall, being strongly structured, can only be flexed so far. And so at some point, no matter how strong the turgor pressure is, water can no longer physically flow into that cell. So not only do you have osmotic pressures here, but you have some turgor pressure playing a role in it as well in how much water can enter in. What happens if a bacterial cell, like I guess this is a bacillus, so let's say Bacillus subtilis, is stuck in a hypertonic solution relative to the cell? So if the solution has, let's say, 6% salt, and inside of the cell it's about 0.9%, what's going to happen then? What's that, Caitlin?
When that salt is the water's going to be into it.
So if the solution outside has a higher concentration of salt than inside,
And the water's going to go out.
good. But that's okay. So you hit the first part. Water's going to move out. Red blood cell, definitely. They crenate. They kind of almost look like spiky by the time that they've lost their water. Do bacterial cells necessarily shrink? And the answer here is they might change a little bit, but largely that cell wall, again, holds up. It helps to maintain the three-dimensional structure of these cells. Instead, what we see is something that's more akin to what we find in plant cells, where you lose some of that turgor pressure and you start to get plasmolysis. And what plasmolysis is when the membrane pulls away from the cell wall. Because of that loss of water, the cell tries to equilibrate across that water movement outside of the cell will shrink the cytoplasm, will shrink the volume of the cell or shrink the surface area that cell membrane is covering, but yet the cell maintains roughly its shape. Erica?
Are we still talking about just gram-positive?
We are talking just about gram-positives. Yes, thanks for asking that. On to that point, Erica, how would this change with gram-negatives?
I would guess, and maybe wrong, that it still wouldn't change shape as much as like the eukaryotic red blood cell. But I would say that it would be easier. I guess if we're
So let's go to your first point real quick. Why do you think that it would that a gram-negative would still maintain structure more than a eukaryotic cell?
because it does have some.
Okay, good, perfect. And if this were a question on an exam, we know gram-positives would gram-negatives be affected? And if you said no, they've got a cell wall that helps to hold the structure, great. You're using evidence that we've talked about in the class to support your answer. Perfect. You might say, where I think you're going with this is, well, it's got a cell wall, but it's thinner, right? And that thinner cell wall may be a bit more susceptible to losing its shape, or in a pure water sample, those gram-negatives may be more likely to actually lyse, because since that peptidoglycan is a little bit thinner, maybe it doesn't hold on to that turgor pressure as well. That would be a great answer too. I honestly don't know the answer off the top of my head. I guess we'll have to go experiment on it to figure it out, since that's part of science. All right. Any questions on these principles? And I also want to make sure real quick that we're good with facilitated, active, and passive transport. I know that there's one slide that probably people went literally WTF, right? So that would be this slide here. And then they were like, wait a second, do we need to know all these? And the answer is no. Okay. These are 3 examples of active transport. What do they all have in common? Or what is one commonality? That is true. That is a key feature of active transport, is typically we find active transporters working against the concentration gradient. We also find that then, conversely, passive and facilitated transport go with the concentration gradient. So go from high to low concentrations. These transporters are used particularly for molecules that might be trace nutrients, ones that are at really low concentrations in the environment, or ones that are for molecules that are essential for life. So, for example, iron, Fe2+ or Fe3+, is a limiting nutrient to most bacterial cells. It's required for life. It's required for enzyme function, and that's something we'll come back to. But oftentimes, bacterial cells are scavenging for iron. And the way that they do is they release these molecules that will bind up to the iron, and those molecules end up finding their way back to the cell just by random motion. And the cell then can actively transport the iron across the membrane and accumulate it inside of the cell. And so that ensures that there's a constant higher concentration of iron inside of the cell than there is outside in the environment. The group translocation one up here is an example of how glucose gets captured in the cell. So in that example, phosphoenol pyruvate, or PEP, it's one of the last molecules produced in glycolysis. It will transfer its phosphate, it's a high-energy phosphate, onto glucose as it's transported across the membrane. That physically changes the structure of glucose enough so that it can't be transported back out of the cell. It essentially locks glucose into the cell at that point. Okay? So these are tricks that cells will use to accumulate some of these limiting resources. Obviously, everybody's going to be familiar with ATP as an energy source. How does the top example differ? Derek? Good. It's using a charged ion, and that one is going with its concentration gradient to pull across something that's going against its concentration gradient. So we can see some of that coupling as well. Let's take a look at an example, one that actually we will come back to in, say, 2 weeks from now. More like a week from now. I take that back. All right. I'm going to draw. You feel free to draw too if you want. This is like Science Pictionary. Anybody know what this is? I'll keep drawing until somebody figures it out. Ah! Okay. Anybody know what this is yet? Yeah, it's the electron transport chains. This is a great example of transport systems, okay? And I'll just simplify this for now. Okay. what I want you to do is to figure out for me, I'm going to put one more piece of evidence in here. So pH. The bottom half is cytoplasm. The top half, in the case of bacterial cells, is between the cell membrane and peptidoglycan, or that periplasmic space in gram negatives. Okay. What I want you to do for me is to determine what kind of transport is being used here. Okay, so in particular, I want you to focus on this one. I'll label it B. And we'll say this one. I'll label it A. You tell me what kind of transport is occurring and what evidence are you using to support those assumptions. Okay, I'm going to give you guys about 3 to 4 minutes to work through this on your own, with somebody else. Feel free, either way is fine. All right, go at it. The answer, in theory, is factually correct. Okay? So, for example, what's one thing that's different between a gram-positive and a gram-negative? Austin?
So, like, the gram-negative has, like, a cell membrane.
Okay.
Peptidoglycan and other cell membrane.
Okay.
The gram-positive is the peptidoglycan.
And, Meg, what's true about that outer membrane?
The outer membrane is harder to get into, so then it's harder, the penicillin, it's harder for it to get into, to like get to the peptidoglycan.
Okay, so you guys are focusing on the peptidoglycan here as the target. Why? What are you using to it absorbs the penicillin?
Absorb the water.
Ah, okay, so you're saying because these cells bursted, because you're saying here that the 0.1% NaCl is hypotonic relative to the cell, water's going to rush in, that results in the cell bursting somehow. And that means then what has to have been weakened? The peptidoglycan, okay? That's very logical. Additionally, the outer membrane is very logical as well, because what have we said about the outer membrane? Selectively permeable. Doesn't allow for larger, bulky molecules necessarily to get through. Prefer molecules that are more hydrophobic rather than hydrophilic can get through that outer membrane. And guess what? That's exactly how penicillin works. That's why it works on gram positives but not gram negatives. So, what I'm looking for in these answers are putting together. That one right there involved basically this whole chapter minus transport proteins and some of the other structural things we haven't talked about, but it combines 3 or 4 different objectives from this chapter. Okay? I love essay questions like that because, one, I actually get a sick pleasure out of writing essay questions. I don't know why. But the other thing is it really shows me what you know, more so than just asking what fill in the blank type word. That's limited in its scope of what you understand and your level of thinking. I think those essay questions do a lot better. With essays, I'm a huge believer in partial points. Okay, so if that one were like a 10-point essay, you could get 0 points all the way up to 10 points, anywhere in between. Okay? Doesn't have to be perfectly correct. Needs to be logical, needs to be consistent message, flow, and factually, in theory, needs to be correct. All right. Okay, so what I'm going to finish up then and I've kind of hit on some of this throughout the day, so we'll probably get out of here a couple of minutes early, but wanted to just make sure to hit on some of the comments that you made. Just so you know, I was having a glass of Cabernet while I was looking at these. Not because it was that disappointing, just because it was a long day. All right. One of the the things that I think I want to reiterate here is, what are my expectations? And the reason why I say that is because a lot of people commented on things like, the mini lectures are good. I know there's some technological issues. I'm still trying to work those out, and I completely understand that can be frustrating. So one of the things that I'm doing is putting these up on YouTube as well, so you can access them another way if you want to. But those are really, I kind of feel like that's the starting point for everything we're going to talk about in class. So if you're not getting a chance to look at those mini lectures ahead of time, the class is it could be very challenging to follow, okay? And in part, that's by design. I'm not trying to make it challenging in here for somebody to follow, but really what I want to do is work on thinking through these problems. I can lecture from sitting next to my dog and on a video, and you'll hear him at some point. He barks at one of the videos, but that's not something that any of us physically need to be here to do, right? 8 o'clock in the morning for a lecture is not the best time of day, and so I don't like to just sit here and talk. And so that's why I put those up beforehand. The second thing is use your resources, okay? And my one concern, I heard a lot of people saying, like, I'm keeping up with the homework, great, I'm keeping up with the mini lecture.